We're going to talk about electron microscopes and scanning probe microscopes in this lecture. The electron microscope is different than the light microscope because it's going to use an electron beam to illuminate the specimen rather than a beam of light. And the electron beam has a much shorter wavelength than the light does. And if you remember, the shorter the wavelength, then the higher the resolution. So that's going to be the advantage of the electron microscope over the light microscope. So it has a much higher resolution than the light microscope does. So it's going to give a much sharper, more detailed picture than the light microscope does. So some of the electron microscopes, you can actually see down to an organic molecule. So it has very, very high resolution, where remember the light microscopes have a much lower resolution and you can only see down to about a bacterial cell and that's it. But the disadvantage of the electron microscope compared to the light microscope is that you can't look at any living cells. And that's because of the way that the cells are prepared to look at them under the electron microscope. That process will kill the cells, so you can't look at any living cells with an electron microscope. This just shows the limits of resolution for the different types of microscopes. So the light microscopes, the resolution is much lower, and so you can see down to about the bacterial cell. And then anything smaller like a virus, you can't see that. So it just gets all blurry and you can't see that the viruses are there. But with an electron microscope, you can actually see down to the size of an organic molecule with some of them. So they have a much higher resolution. And this just shows the difference between looking at bacteria with a light microscope over here on the left, and then here's looking at the same bacteria but using an electron microscope. So you can see the resolution is much higher along with the magnification, and so you can see a lot more detail here. It's a lot sharper and much more clear image, so you can see a lot more parts inside that bacterial cell. There's two different types of electron microscopes. We have transmission electron microscopes, which are abbreviated TEM, and that you're looking at a cross section. So the specimen is going to be sliced into a very thin section and you're going to be looking inside of the cell. And then we also have scanning electron microscopes, or abbreviated SEM. And that one you're looking at a three-dimensional image of the surface of the cell. So this just shows the difference between the light and electron microscopes. So the light microscope is over here on the left, and I'm looking at a paramecium here. And in the middle, this is a transmission electron microscope. So this one, we've cut our paramecium into a thin section, and then we've coated it with some heavy metals. And then we're going to shine a beam of electrons onto our specimen, and that will go right through the thin cross section. And instead of going through glass lenses to help focus and magnify the image, we're going to use instead some magnets to help focus the electron beam. And then that will hit the screen here, and then the computer will make the image. And so with our transmission electron microscope, we're actually looking inside of the paramecium here, so we can see the structures inside there. And then with the scanning electron microscope, we're taking a look at the surface of the paramecium, and so you get these really cool three-dimensional images of the cells. And these are also prepared by rolling the cells into heavy metals. and that will also kill the cells. So when the electron beam hits the cell coated in the heavy metal, that'll knock electrons off of the heavy metals, and then that will then hit the electrons will then hit the screen, and then that will create the image. So this is just showing the transmission electron microscope, what it looks like. So it's a pretty big structure here. And here's a picture from a transmission electron microscope of a bacterial cell. And again, they've sliced it into thin sections, and so we're actually looking inside of the bacterial cell. This structure that's inside of the bacterial cell, this is called an endospore, which we'll learn about in chapter 3, and we'll talk about what that is. And this is just a comparison between the light microscopes and the transmission electron microscopes. All you need to study on this table is just what we've talked about. So the magnification on the light microscopes, we can only go up to about 1000x. So with our ocular lens being 10x and our objective lens 100x, so it's about 1000x would be our maximum magnification there. So with our transmission electron microscope, a lot of them are going to be over 100,000x, and some of them have even gotten to be up to 250,000x, so they're pretty powerful. And then the resolution. This is talking about how close do these two different points have to be, so you can see them as separate, distinct points. So with the light microscopes, as long as they're 0.2 micrometers or 200 nanometers apart, you can tell that there's two separate points there. So that's pretty low resolution. And then with the transmission electron microscope, much higher resolution. So as long as the 2 points are 0.5 nanometers apart, you can tell that there's two distinct points there. And then just the other thing that we talked about, the source of illumination for light microscopes would be light, and then for the transmission electron microscopes, that would be an electron beam. The other type of electron microscope we have are the scanning electron microscopes. And this one, we're going to be taking a look at the surface of the cell, and you get these nice three-dimensional images, which are pretty cool. The computer allows you to color them. They're actually black and white images, so they're really not that color. And you roll these specimens in heavy metals when you prepare them, and that will actually kill them. So again, you can't look at any living specimens with an electron microscope, so that would be their disadvantage. And this is just showing you how the scanning electron microscopes work. Here's the electron gun shooting out the beam of electrons. And then you've got these electromagnets that will help focus the electron beam. Hits the cell coated with the heavy metals, and that'll knock the electrons off, and that'll hit the detector, and that'll make the image. And if you're interested, SLIC actually has a scanning electron microscope, and you can actually take a class and actually learn how to use it, which would be pretty cool. These two pictures are of the same microorganism, Giardia, one of our protozoans, a eukaryotic microorganism. The picture that is on the left is looking at a cross-section, and we're looking at the inside of the cell. So this was made by a transmission electron microscope. And then the picture on the right, we're looking at the surface of the Giardia cell. So this one was made by a scanning electron microscope. And you can see that it moves around by some flagella. And then on this end over here, there's actually a suction cup. And it actually will stick to your intestinal wall with a suction cup. And that's how it stays in there and doesn't get pooped out. So that's kind of gross. Okay, and the last type of microscope we have, so besides the light microscopes, an electron microscope, this third type of microscope we have, these are called scanning probe microscopes. and their name says what they do. So they're going to use a probe to scan the surface of the specimen, and they create this really cool three-dimensional image. And there's two different types of scanning probe microscopes. So there's scanning tunneling microscopes and atomic force microscopes. So the scanning tunneling microscope, it has very high resolution, and what it does is it has a very fine tip probe. The tip on the probe is actually 1 atom thick, so it's very, very thin. And the probe will be a certain distance above the specimen that you're looking at. And here we're actually looking at a DNA molecule. So the resolution is so high that you can actually see down to the atom level. And so you can actually see the atoms here and making up this DNA molecule, which is really cool. And you can actually see the double helix here. So what they do with this probe is they will keep it a certain distance above the specimen so it actually doesn't touch it. And then they'll run a current, electric current, between the probe and whatever it is you're looking at, so like this DNA molecule. And then they will run the probe over the specimen that you're looking at with that electric current in between and keep it at a certain distance. And then the computer will keep track of the movements of the probe and then put together the picture for you. So that's the scanning tunneling microscope. So very, very high resolution, and you can actually see the outlines of the atoms here. So that's pretty cool. Okay, and then the atomic force microscope. This is the other type of scanning probe microscope. So this one's a little bit different. It doesn't use an electric current. So this one, the probe, the tip is actually going to touch the specimen, so it's going to rest right on it, and there's not going to be any electric current involved, and so the tip will just trace right over the specimen, whatever it is you're looking at, and then there's a laser that will hit the top of the probe, and then the computer will pick up the movements as the probe goes up and down over the specimen, and then the computer will put together the picture. And here are the pictures that we can get with the atomic force microscope. So here's one over here on the right. These are some proteins that are produced by a bacterium called Clostridium perfringens. This bacterium causes food poisoning, and it produces these toxins that are made out of proteins, and they poke holes in your intestinal cells, and that causes them to leak fluid out and gives you bad diarrhea, and that causes the food poisoning. And then this picture over here is another protein. This one was made by a scanning tunneling microscope, and this protein is used by E. coli to actually repair its DNA if it's been damaged. So these two microscopes have very, very high resolution. You can see these organic molecules, so they're pretty cool microscopes. The scanning tunneling microscope uses an electric current, whereas the atomic force microscope does not. So that's kind of the difference between the two. And then the atomic force microscope, Slick actually has one of those, and you can actually take a class to learn how to use that one if you're interested.